Very good morning to all of you. So today's our topic of discussion is that of mucopolysaccharides or glycosaminoglycans. This topic is very important topic and must read topic for PG as well as for UG students both. Because in carbohydrate chemistry, the question is frequently asked on this topic. Okay, so let's understand the topic. What is mucopolysaccharides or glycosaminoglycans? Okay. So as you know, the carbohydrates are what? The carbohydrates are classified into three. That is of monosaccharides, then your disaccharides, then your oligosaccharides, and then polysaccharides. Okay, as you know it before from the classification of your carbohydrates. Okay, so mono, di, oligo and your polysaccharides. So these mucopolysaccharides or glycosaminoglycans are categorized under what classification? These comes under your polysaccharides. What are polysaccharides? When there are more than 10 monosaccharides units. Okay, monosaccharides units. So when there are more than 2 monosaccharides units are there, that are categorized under your polysaccharides, and these polysaccharides are further categorized into your homopolysaccharides and your heteropolysaccharides. Okay, so what is the term homo? Homo means same. So in homopolysaccharides, the repeating units of the monosaccharides are same, while hetero means different. So in heteropolysaccharides, the repeating units are different. Okay, so these glycosaminoglycans comes under your this heteropolysaccharides. So what are the examples of your heteropolysaccharides? So when you will read these examples of your heteropolysaccharides, then it includes your agar, agarose, pectin, actins, gums, and your GAGs. Okay, so heteropolysaccharides are those polysaccharides which consist of two or more than two different kinds of monosaccharides unit. And examples of this heteropolysaccharides is that of agar, agarose, your pectin, your gums, and your glycosaminoglycans. Okay. So these, now what you will write in your exam exactly when question asked as a short note on mucopolysaccharides. So first, how the question is asked in the exam, the question is asked like a short note on the glycosaminoglycans on mucopolysaccharide, or if long question will come, then the long question can be asked as a what is heteropolysaccharides, give examples of heteropolysaccharides, 1 define your mucopolysaccharides or glycosaminoglycans along with their disorder. It is the pattern of your long answer. Otherwise short note also asked as a, if 10 marks question will be there, then mucopolysaccharides define along with examples and what are the different disorders or what is mucopolysaccharidosis. Okay, so this is the format of question which is asked on this topic. So what you will write? First you will write the features. What are the features of this mucopolysaccharide? So first these are what? These are your heteropolysaccharides. These are your heteropolysaccharides. Okay. Alright. So these are your heteropolysaccharides. And why called as mucopolysaccharides? Why the term mucopolysaccharides given to these? Because why this term is given? Because they are first discovered in mucin. Okay, because these were first discovered in your mucin. Mucin is what? Mucin is a mucus secretion. That's why this term mucopolysaccharides is given to these substances. Alright. So question can be asked in the viva, why these mucopolysaccharides are known as mucopolysaccharides? Because these were first discovered in your mucin. That's why these are known as mucopolysaccharides. Okay. The first point is that these are the heteropolysaccharides. These are called mucopolysaccharides because they were first discovered in your mucin. Next is they are long and unbranched. Okay. unbranched. So no branching is there in these mucopolysaccharides. They are long in nature and they are unbranched. Right? They are long, repeating units are there, but they are unbranched. Okay, unlike glycogen, which is very branched molecule. Okay, so heteropolysaccharides first discovered in mucil, long and unbranched. Fourth point is they are what? Negatively charged. What kind of charge they carry? They are negatively charged. Why they are negatively charged? Because they carry your various sulfate groups are there, phosphate and your acetate. And all these groups are your what? All these are negatively charged groups. Sulfate, phosphate, acetate, all these carry your negative charge because these sulfate, phosphate and acetate group are attached with these GAGs. That's why they are overall charge on them is negative. That's why they are overall negatively charged. Now, because of this negatively charged, what they will ultimately do? They will repel each other. Okay, because what is these are the polysaccharides, so they consist of repeating units. No? So repeating units is there, and because of these, in these repeating units, either there is a sulfate or there is a phosphate or there is an acetate on adjacent position. So because of that, these negative charge, they will repel each other. They will repel each other, and because of this repellent action, because they will repel each other, and because of it, they are slippery in nature. Slippery in nature and viscous. And your viscous. Got it? So question can also be asked, why the glycosaminoglycans or mucopolysaccharides are slippery in nature or viscous in nature? Because of the containing the negatively charged groups like sulfate, phosphate and acetate. Because of that group, they are overall negatively charged. So because of they are overall negatively charged and repeating units are there. So that molecule ultimately repel each other. There is a repulsion in between negatively charged and negatively charged molecule, and because of that repulsion, they are slippery in nature and they are viscous. Okay, so these are the characteristics. Okay, now comes on the composition of this mucopolysaccharides. What is the composition? Okay, so what is composition? So as you know, it is a heteropolysaccharide. That means they are made up of what? They are made up of your two different units. They are made up of two different units. Okay. So these are made up of what? These are made up of your two different kinds of unit. One is that of your amino sugar is there, and another is your uronic acid. Okay, so these GAGs are made up of your amino sugar and uronic acid. Amino sugar and uronic acid is their composition. Now this amino sugar can be your glucosamine, or this amino sugar can be your galactosamine. Okay. So amino sugar can be your glucosamine, or amino sugar can be your galactosamine. Similarly, uronic acid can be your glucuronic acid or your iduronic acid. Alright, so what is the composition? The composition is that they are made up of two different units. First is that of amino sugar and uronic acid. So in all the glycosaminoglycans, the amino sugar and uronic acid is there, and amino sugar could be either it could be glucosamine or galactosamine, and uronic acid may be glucuronic acid and your iduronic acid. Now remember, one exception is here, that is of your keratin sulfate. Keratin sulfate is only GAG which don't have your uronic acid. Okay, so always remember this is a very, very, very important MCQ asked multiple times in many exams and for your viva also. That which glycosaminoglycans or which mucopolysaccharide don't have uronic acid. So remember, keratin sulfate is the only GAG which don't have a uronic acid. Okay, in keratin sulfate neither there is a glucuronic acid nor there is a alduronic acid. Okay, I will discuss later what is the composition. So for your MCQ and viva, you must remember this is keratin sulfate is the only GAG which don't have your uronic acid. Alright. Now we will read individually the different type of your GAGs. So first is your hyaluronic acid. So what is the example of these GAGs? The example of these GAGs are that of your hyaluronic acid, chondroitin sulfate, dermatan sulfate, keratin sulfate, heparin and your heparin sulfate. Okay. We will teach it individually all these. So first is your hyaluronic acid. Okay. First GAG is your hyaluronic acid. So what is the composition of this hyaluronic acid? It is made up of your N-acetyl glucosamine. N-acetyl glucosamine. It is hyaluronic acid made up of the amino sugar is N-acetyl glucosamine and there is a glucuronic acid. Okay, so composition is that of N-acetyl glucosamine plus glucuronic acid is there in your hyaluronic acid. Okay. So now what are the important features regarding this hyaluronic acid? That all GAGs are sulfated except your what? Hyaluronic acid. Okay, so remember all GAGs except of this hyaluronic acid, all glycosaminoglycans are sulfated. Okay, the question can be asked in an MCQ, which of the following GAGs is not sulfated? The answer should be your hyaluronic acid. Okay, so this is the exception that hyaluronic acid is not sulfated. Second thing is that what is its function? It is viscous in nature. As you know, all GAGs are viscous in nature. So present it where? It is present in your synovial fluid. And your vitreous humor of your eye. Synovial fluid and your vitreous humor of your eye. So where this hyaluronic acid is present, it is present in your synovial fluid and it is present in your vitreous humor of eye. Okay. So what is the function? It will provide what? It will provide viscosity, as it is viscous in nature and act as a shock absorbent. So what is the function of this hyaluronic acid? It provides the viscosity to the organ, like it is present in between the knee joint. Hyaluronic acid present. That's why it provides the viscosity, and because of viscous nature, it acts as a shock absorbent. Okay. So this is the hyaluronic acid, its composition, and where it is present here in your synovial fluid and vitreous humor of eye. What is its function? It provides viscosity and act as a shock absorbent. And composition, it is composed of N-acetyl glucosamine and your glucuronic acid. Alright. Now comes our second GAG. Second is your chondroitin sulfate. Second GAG is of your chondroitin sulfate. Chondroitin sulfate. Okay. So in one of my previous video of mnemonic of day, I already shared the trick how you will remember the composition of your chondroitin sulfate and your dermatan sulfate. If you don't watch that, please watch that. Here again I am just repeating that how you will remember that I shared the mnemonic that when you will go in a gala night or gala dinner, then there you will listening the music where you will listening the music on the CD. Okay, so in chondroitin sulfate, CD, C for chondroitin sulfate and D is your dermatan sulfate. So in chondroitin sulfate and your dermatan sulfate, what composition? Gala night, you can link it with the galactosamine. So when you will go in a some like gala dinner and you listening in the music on the CD, okay, so chondroitin sulfate and dermatan sulfate, the composition of your an acetyl galactosamine. Okay, so these two, that is chondroitin sulfate and dermatan sulfate, is only GAGs which consist of an acetyl galactosamine. Otherwise, all other GAGs having an acetyl glucosamine. Okay, so what is the composition? The composition of this chondroitin sulfate is that of an acetyl galactosamine and acetyl galactosamine plus your glucuronic acid. And acetyl galactosamine plus your glucuronic acid. Okay, so this is the composition of your chondroitin sulfate. Now, what are the features? Chondria. Chondra means what? Chondra is a Greek word, and chondra in Greek word chondra means your cartilage. Okay, so chondra means what? Chondra means your cartilage. So it is found in where it is found? This chondroitin sulfate is found in your cartilage, bone, and in your tendon. Okay, remember chondroitin sulfate, chondra means cartilage, so these are present in your cartilage, bone and your tendon. And composition is, it is made up of your an acetyl galactosamine plus your glucuronic acid. An important thing that you remember, chondroitin sulfate is your most abundant GAG. Okay, most abundant GAG. This is important for your MCQ and Viva. That is, which glycosaminoglycans is the most abundant. So remember, chondroitin sulfate is the most abundant. That is, it is the most abundant all of other GAGs. So if question is asked, which of the following is the most abundant GAGs, the answer should be your chondroitin sulfate. Okay, so this point you must remember regarding your chondroitin sulfate. So it is present in cartilage, bone and tendon. So what is its function? Is provide your shape and your structure. Okay, provides your shape and structure. So once again, what is chondroitin sulfate? So remember, chondroitin sulfate and dermatan sulfate is only GAG which consists of your an acetyl your galactosamine. Galactose, that is galactose sugar is there in amino sugar. That is an acetyl galactosamine, while all other GAGs having a your glucosamine. So composition, it is made up of an acetyl galactosamine plus uronic acid is that of glucuronic acid is there, and it is most abundant GAG, and chondra means cartilage, so it is present in your cartilage, bone and tendon, and its function is to provide shape and structure to the concerned organ. Okay, now comes another GAG, that is of your dermatan sulfate. Next GAG is your dermatan sulfate. Dermatan sulfate. Okay, dermatan sulfate. So what is its composition? It is made up of your, again it is made up of your an acetyl galactosamine and acetyl galactosamine plus it is made up of your iduronic acid. Okay, now how you will remember which uronic acid is present is where. Remember, gala night having a CD, you can buy this pneumonia, you can easily remember that chondroitin sulfate and dermatan sulfate, what it both these contain an acetyl galactosamine. Now confusion in between your uronic acid. So remember, dermatan is having a D, okay, and iduronic acid also having a D. So remember, in dermatan sulfate there is a iduronic acid, while in chondroitin sulfate there is a glucuronic acid. Okay, so D comes in dermatan. So remember, D from D you can remember D dermatan iduronic acid. D is dominating here. So in dermatan sulfate iduronic acid is present, while in chondroitin sulfate glucuronic acid is there. And your amino sugar in both chondroitin and dermatan is that of an acetyl galactosamine. Okay. Now next point is that derma. Derma means what? Derma means what? Derma means your skin. Derma means your skin. So where it is present? It is present in your dermatan sulfate is present in your skin and your heart valves. Heart. Okay. And what is function? Function. What is its function? This function provide your shape and structure to the organ, like your chondroitin sulfate. Okay, so dermatan sulfate composition is made up of an acetyl galactosamine and hydroronic acid. Derma means skin, so it is present in your skin and your heart walls. And what is this function? Like chondroitin sulfate, similarly it provides the shape and structure to the concerned organ. Okay, so this is your dermatan sulfate. Now comes next our is that of keratin sulfate. So next GAG is our that of your keratin sulfate. Keratin sulfate. Okay. So keratin sulfate is what is composition? Keratin sulfate is made up of your an acetyl what? Glucosamine. As I told you, only chondroitin and dermatan having a galactosamine. Otherwise, all GAGs having your what? Glucosamine. So the amino sugar is in keratin sulfate is that of an acetyl glucosamine. And as I told before, this is the exception. This is the only GAGs which don't have any uronic acid. Okay, so neither it have hydronic acid nor it have glucuronic acid. Besides that, it having galactose in its composition. Okay, so this is the most important MCQ. Okay, so composition is it is made up of an acetyl glucosamine plus galactose. Because important thing is that you must write that no uronic acid present in your keratin sulfate. Okay. So there is no uronic acid. Besides uronic acid, galactose is there. And normal your amino sugar is an acetyl glucosamine. Okay. And this keratin sulfate is present where? This keratin sulfate present in your cornea and your cartilage also. So this keratin sulfate is present in your cornea and your cartilage. And what its function is provide corneal transparency. Transparency. That means it provides transparency to the cornea. So this is the function of your keratin sulfate. So remember the important point keratin sulfate. Keratin sulfate is only GAG which don't have any uronic acid. What is composition? It is made up of your N-acetyl glucosamine plus your galactose. In spite of their uronic acid, galactose is there and it is present in your cornea and cartilage. And what is its function? It provides transparency to the cornea, or you can write corneal transparency is its function. Okay. Next is that of heparin. Next GAG is that of heparin. So heparin. Heparin is made up of what? It is made up of your N sulfate glucosamine plus glucuronic acid or hydroronic acid is there. So the composition of this heparin is that here the glucosamine in heparin is sulfated form. So composition you will write N-sulfate glucosamine, and the uronic acid either there may be glucuronic acid is there or there may be hydroronic acid is there. Okay. So what is the important points in heparin? It is only GAG which is intracellular. Okay, this is the important point. It is only GAG which is intracellular. As all other glycosaminoglycans, remember, as all other glycosaminoglycans are present in extracellular cellularly, because the main function of glycosaminoglycans is that these glycosaminoglycans combine with the protein and they will form proteoglycans. Okay, so when glycosaminoglycans will combine with the protein, then they will form your proteoglycans, and that proteoglycans along with your collagen and other molecules forms your extracellular matrix, and that is the main function of your this GAGs. And so all the GAGs are extracellularly present, but heparin is exception. It is also present in your intracellular. Okay. So where it is present? It is present in your. It is present in, as you know it before, heparin acts as an anticoagulant. Okay. So it is present in your what? Blood. In your mast cells, it is also present, and in your lungs. Okay, so it is present in your blood, it is present in your mast cell, it is present in your lungs. Okay, and what is its function? It acts as a anticoagulant. Okay, so its function is it acts as a anticoagulant. Okay, and another function of this heparin is it also helps. It also helps in release of lipoprotein lipase. Lipoprotein lipase enzyme. Okay, it also helps in the release of your lipoprotein lipase enzyme from endothelium. Okay, so lipoprotein lipase enzyme, which is present on the endothelial surface, and with the help of this heparin, this lipoprotein enzyme will easily release. Okay. This lipoprotein lipase you will study detail while you will study your chylomicrons metabolism. Okay. So now at this level you have to remember what is the function. What is the function of your heparin? So the function of heparin is that of it acts as an anticoagulant and it helps in the release of your lipoprotein lipase enzyme from the endothelium. Okay. So let's again, heparin is what? The composition, it is made up of N-sulfate glucosamine. Remember the glucosamine having here the sulfate group. So composition is N-sulfate glucosamine plus either there may be glucuronic acid or there may be hyaluronic acid, and it is the only GAG which is present intracellularly. Otherwise all GAGs are extracellularly, and it is present in your blood, mast cell and lungs. And what is its function? In blood it acts as an anticoagulant. Besides that, it also helps in the release of your lipoprotein lipase enzyme from the endothelial surface. Okay. Next and last is that of heparin sulfate. Next GAG is that of heparin sulfate. Heparin sulfate. Okay, heparin sulfate. So heparin sulfate having the similar composition as that of heparin, because the heparin is also there. Heparin. So composition is similar to heparin. Heparin. What difference? The difference is except it is less sulfated. Although the term sulfate in heparin sulfate is present, but remember the heparin sulfate is less sulfated as compared to the heparin. Okay, so what is this composition? Heparin sulfate has a composition similar to that of heparin. That is, it also having N-sulfate galactosamine and sulfate glucosamine plus your either glucuronic acid or hyaluronic acid. But as compared to heparin, it is less sulfated and more acetylated. That means the sulfate group linked to the your glucosamine is less in case of heparin sulfate and acetyl group is more. Okay. Otherwise composition is same. Okay. Now what is the function of this heparin sulfate? Where it is present? It is present in your basement membrane. It is present in your basement membrane of your, where it is present in basement membrane of your kidney. In Bowman's capsule, the basement membrane of Bowman capsule is made up of your heparin sulfate, and this is the reason why albumin is not present normally in the urine, because heparin sulfate is negatively charged and it forms the boundary of your basement membrane of your Bowman capsule, and because of this negatively charged group of heparin sulfate, the ultimately charge on the basement membrane of your Bowman's capsule or kidney is negative. So when albumin comes in your glomerular filtrate, albumin is also negatively charged. So negatively charged basement membrane repel the negatively charged albumin. That's why normally albumin is not present in the urine. But when there is a suppose glomerulonephritis there, when there is an inflammation, then the structure of this basement membrane is erupted. So when the whole structure of this basement membrane, like in example in the diseases of glomerulonephritis, interrupted, so negatively charged loss, and because of loss of that negative charge, now albumin is not repelled. So albumin is filtered out through the glomerular filtration. Okay. So remember, it is present where? It is present in the basement membrane of your kidney. Okay. And what is function? Its function, as I told you, its function is prevent loss of albumin in urine. Albumin in urine. Reason is similar, that because negative charge repel each other and albumin carry your negative charge. So albumin cannot be normally filtered into your glomerular filtration. Okay. So this is all about all your GAGs. So GAGs are what? These are also known as mucopolysaccharides. Why these are known as mucopolysaccharides? Because these were first discovered in your mucin, and mucin is your mucus in nature. Okay. Next important, what are the examples? Like your hyaluronic acid, chondroitin sulfate, dermatan sulfate, keratin sulfate, heparin, and your heparin sulfate. Important thing is that which glycosaminoglycans don't have uronic acid? So answer is your keratin sulfate. Okay. Next important, most abundant GAG. So most abundant GAG is your chondroitin sulfate. Next important question, MCQ in that, which of the following GAG is found intracellularly? So intracellular only GAGs is your that of heparin. And you should remember all the composition. Next question is that, which of the following GAG contains of composition as an acetyl galactosamine? So remember galanite with the CD. By this mnemonic, so chondroitin sulfate and dermatan sulfate is the only GAG which consists of your an acetyl galactosamine, while all other GAGs have an acetyl glucosamine. Okay, so this is all about your mucopolysaccharides, and in next video I will share the disorders of the mucopolysaccharides, that is known as your mucopolysaccharidosis. So thank you for today.